Hi, today we shall be discussing the structure of the tRNA molecule. So, this is the classical clover leaf shape of a tRNA molecule, and it has D loop, anticodon loop, a T loop, and an acceptor arm. This is an acceptor arm. This acceptor arm is formed by the nucleotide pairing between the 3 days, the 3 days end and the 5 days end of the tRNA. The 3' end has a hydroxyl group and the 5' end has a phosphate group, and this 3' end has ACC acceptor ACC this basis, and this is the area of the tRNA on which the amino acids are loaded up by amino-acyl tRNA synthetase. So, what are the functions of the other parts of the tRNA? The D loop, along with the acceptor arm and the anticodon loop, they all help the amino-acyl tRNA synthetase, which is the enzyme which loads the amino acids on the tRNA. So all of these, the D loop, anticodon loop, and the acceptor arm, they help the amino-acyl tRNA to recognize the amino acid binding site of the tRNA. Now, basically, what is tRNA? tRNA is there are three forms of RNA: mRNA, rRNA, and tRNA. tRNA is the non-coding form of RNA, which helps in bringing amino acids to the mRNA molecule which is being transcribed. So an mRNA molecule is transcribed by a ribosome. So the mRNA has a codon, and that codes for a particular amino acid. So that amino acid has to be brought to the mRNA. How this is going to happen? The codon of the mRNA binds to the anticodon on the tRNA, and this anticodon, D loop, and acceptor arm, via the binding of the codon, they recognize which amino acid is to be brought, and amino-acyl tRNA synthetase binds that amino acid through to the 3' end of the tRNA. So this is how the amino acid is brought to the mRNA, and subsequently the whole of the mRNA molecule is transcribed. The D loop function we have already discussed. It is also known as dihydrouridine group because it has a base called dihydrouridine, and there is a T loop which is also known as T. This is a weird base. So this T and this symbol, it stands for ribothiamidine, pseudouridine, and cytidine. Ribothiamidine, pseudouridine, and cytidine. What the T loop does it, T for T, if we remember, T ties the tRNA to the ribosome, because we know that the anticodon loop of tRNA binds to the mRNA, and the mRNA is bound on the ribosome. So the tRNA has to bind to the ribosome. So this binding of tRNA to the ribosome molecule, the ribosome, is actually achieved by the T loop. So this is basically the structure of a tRNA and the function of various parts of tRNA.